and consultant neurologist. And we have Professor Zoe Kourtzi, who is a professor of cognitive computational neuroscience. And they're going to speak to AI for better brain health, turning the hype into reality in dementia.
Great. Thanks. Thanks for the introduction. It's a real pleasure to be invited to speak at this meeting. I think it's been a fantastic day. I've really enjoyed it so far. And we're going a little bit more back to the clinical side of things now. You get me first, and then Zoe will. I'm sort of the warm-up act for Zoe, I think, is the best description. Artificial intelligence, is it just all hype, or is it actually starting to happen? And I'd like to argue that we're starting to be able to use AI meaningfully, particularly for clinical translation. And these are the sort of three things that we're sort of going to focus on, and I'll sort of try and set up, and Zoe will show you how we've been addressing that with one machine learning algorithm and approach. So firstly, you've heard a little bit today about relevant clinical questions from Ben. We'll talk a bit more about that. And the need for real-world data and explainability is critical in clinical applications. So what happens currently when you come to a memory clinic? Well, you see someone like me, and we go through a history and an examination, and we get some additional information from a cognitive test, and from in Cambridge you'll get an MRI scan. In other places you may get a CT scan, or sometimes no scan at all, and we make a diagnosis based on that information. In some centres there are further tests like spinal fluid and PETs, which are available, but they're invasive or expensive. And as Ben pointed out earlier, really this has changed very little over the past 25 years. And there are challenges that we face in the memory clinic. The first is about accurate diagnosis. So from surveys of memory clinics across the UK, we heard about dementia with Lewy bodies earlier. We know that we're only picking up about half the people who should have a diagnosis of dementia with Lewy bodies. For frontotemporal dementia, it's even worse. We're only picking up a tenth of the people that should have a diagnosis based on population prevalence. And even when you have something like Alzheimer's disease, if you have something a bit funny, so you might have psychosis, you're much more likely to get the wrong diagnosis. And this is data from post-mortem studies from the US. So accurate diagnosis is a challenge. Once you've got a diagnosis, most people's question is, well, what happens next? I was in clinic yesterday and got asked this multiple times, okay, well, what's the future hold for us? And particularly the relatives are very keen to be able to plan ahead, know what's coming. And we can tell a little bit from the diagnosis. So if you have semantic dementia, your prognosis is much better than progressive supranuclear palsy, where the prognosis is very poor. But even if you have, say, Alzheimer's disease or frontotemporal dementia, you can see here that your prognosis can vary from sort of 2 to 3 years up to 10 years or even longer. So being able to predict and pick that up is really important. I think Ben Underwood showed really nicely earlier how what a challenge this is, and how he's taken a creative approach to that. So these are some of the clinical questions that we need to address. The other issue, sort of feeding back to Kath Bunbury's talk this morning, is picking people up for intervention and prevention. We looked at the UK Biobank, and we found 5 to 9 years before people got a diagnosis of a variety of different neurodegenerative diseases, we could see subtle changes in their cognition and their day-to-day function. So there's a real opportunity, if we can find something which identifies the earliest stages of these different neurodegenerative diseases, we've got a window of opportunity to intervene when people have a disease, but they're not bad enough yet to come forward for a diagnosis. So on that basis, can AI help? So artificial intelligence is a sort of range of mathematical models, and they try to do that sort of gut feeling that we have as humans, try and classify things based on pattern recognition. And when we started up this sort of study to collect real-world data and apply Zoe's algorithms to this, we got picked up by the BBC, which was nice, and they wrote this story about artificial intelligence may diagnose dementia in a day. Lots of hype, and a lot of my colleagues said, oh, "Well done. We read about you in the Times or the Telegraph or the Guardian." They didn't notice we were front page of the Express. Alistair Cole said, you've done very well to get on the front page of the newspaper, a study that doesn't have any results yet, which is partly true. So is this just all hype? So fortunately, this was a few years ago, and we've got a little bit further now. So just to give you a sort of idea of the range of uses of AI in dementia and neurodegenerative diseases more generally. So this is just what I'm doing in my lab and the different approaches that we've used. And this is very collaborative. This is about multidisciplinary work. And in neuropathology work, we collaborated with Annalise Quagabert and Dave Klenman spoke this morning about touched on some of the work that we're doing, trying to understand progression using this data. And that data comes from AI machine learning assessment of neuropathology slides. We've been looking at brain structure, which uses particularly in progressive supranuclear palsy, and that's using some algorithms which are developed at Imperial. We've looked at diagnostic classification of atypical Parkinsonian disorders with Marta Correa, who's here today, using diffusion tensor imaging and careful selection of features. We looked at pre-symptomatic changes in the UK Biobank using structural brain imaging, and this is using a deep learning approach in collaboration with the computer science department. And we've looked at how we can implement these algorithms into the NHS, particularly in this way called federated learning, which preserves privacy and does data analysis locally rather than centrally. And that's a collaboration with UCL. So these are not the main algorithms that we're going to talk about today, but just to demonstrate that AI and machine learning are transforming the way that we do research. So when it comes to clinical application, there's no shortage of algorithms which have been applied. And Robin Borchert, who's an academic clinical fellow now in radiology, led a review which is part of the Deep Dementia Phenotyping Demon Network working group. And we looked at as many algorithms as we could. There are 255 algorithms that have been applied to neuroimaging looking at either diagnosis or prognosis in dementia. And they all perform really well. None of them have reached clinical practice. And there are a number of reasons for this. First, to say there's lots of things which are encouraging. There's lots of data which is now available, which is allowing us to use more complex techniques like neural networks. There's more data available across different imaging modalities. So people are starting to combine imaging modalities, which is improving the accuracy. But barely any of these 250 algorithms have been validated in an independent data set, and one had been validated unsuccessfully in a real-world data set. So there are still massive barriers to getting this into the clinic. So we've tried to address that issue of real-world data by setting up the quantitative MRI in NHS memory clinics, or QMIN-MC study. This started in Cambridge and CPFT, and a huge thanks to colleagues in CPFT who are by far the biggest recruiters to this study. They just overtook Addenbrooke's memory clinic earlier this year. A bit disappointing, but they've done very well. And we've now got over 700 people recruited to this. We're open at 10 sites across the country, spreading across the whole geography of the country, from sort of Bradford and Leeds in the north down to Southampton and Gloucester. We're looking at making this a more diverse data set in recruiting from King's and from other places in London and Birmingham. When I set this up, I sort of didn't quite realise the power actually of having an unselected memory clinic population, which means we don't just concentrate on one disease. If you look at the open data sets which are available for research, they concentrate on a single disease, and they're very highly selected patients. They don't have any comorbidities. There's no doubt about the diagnosis. They're very unlike the patients who come and see me in the memory clinic. And we've made this data open through the Dementia Platform UK, and there's Hotchkiss here from DP UK, and they've been brilliant at helping us take this data and make it available for anybody to use. So, and if you want to use it, please let me know. So I'll just sort of wrap up my bit there to say, we really need to get the relevant clinical questions right that we want to answer. This just isn't just about diagnosis of Alzheimer's disease versus healthy controls. We need real-world data to be able to properly validate these algorithms, and for clinical use, we need to be able to interpret these algorithms, and that's what Zoe has been working on.
Okay, great. Thank you, Tim. Thank you, Paul and Devila and everybody for organizing and for inviting us for this double act. I think Tim set a really high bar. I'll try to bring it to reality, but I hope with a hopeful end. So let's see, their slides should come up. Okay, wonderful. It's been wonderful actually working with Tim. This is a truly cross-disciplinary project. Please don't take the fact that we cannot coordinate slides as an example of how we work. We work fantastically well together. We have learned millions from each other, and really wonderful to also see early career researchers in our teams working together. So can we use AI for addressing the challenges that Tim put forward? So I'll take it one by one and show you how we try to address these challenges. The first is, can we make AI to be really biologically informed and clinically relevant? So that answers interesting questions that can lead us to translation and adoption in clinical healthcare systems. The second big challenge for us is AI interpretable? Can we actually make sense? Can we understand disease mechanisms? And therefore, by gaining this deeper mechanistic understanding, as we heard earlier, be able to facilitate treatments, interventions. And finally, is it interoperable? I always say dementia, and you know it better than anybody else, dementia is a global problem, it needs a global solution. So it cannot be just a Cambridge solution. It has to apply across the UK, as we saw the communal study with Tim, but also beyond the UK. And I'll try and show you where we have got to in terms of addressing these challenges, our progress so far. So the first clinically relevant question that we want to ask is, can we predict early, and how early we can predict? As Tim explained, 10 to 15 years before we see symptoms, we have dementia in the brain. This is an amazing opportunity in terms of a window where we could potentially make a difference by changing lifestyle factors or with upcoming promising treatments. And that's where we want to be able to make this early and precise patient stratification and prediction. You won't be surprised if I told you this is a healthy brain. Well, this is a brain with Alzheimer's disease. This is what we get from MRI scans. The space we work in is this space. The brain of a patient diagnosed with mild cognitive impairment at that very early stage, by the naked eye, looking at these MRI images, is really hard to tell whether this is an MCI patient, a patient that will develop Alzheimer's disease, or will remain healthy. And that's where we bring in the algorithms to answer this question. And to do this, given that we cannot do it from one single data type, from one single modality, we train models with a wealth of data. And thanks to the patients that, of course, come and join research studies, and they give all of this data. We use biomarker data, blood marker data, we heard from PET scans, we use MRI scans, we use cognition, we use genetics. That's how we train the models. We build the models, though, in a way that then it allows us to remove the most expensive and most invasive markers and make predictions on the data that Tim was talking about, one MRI scan and some cognitive scales. Now, the models are very interpretable, very transparent. We start by giving them features that we have extracted to be biologically relevant. For example, gray matter in entorhinal hippocampal cortex, where we know that Alzheimer's disease will have an impact. We then move on to making really precise classifications, not if somebody is healthy or Alzheimer's. Tim knows this once anybody walks in his clinic, but whether actually somebody at that MCI kind of stage will develop or not disease. And then finally, we care a lot about prognosis and trajectories. Every individual has a different path, a different trajectory. And therefore, we don't stay at this kind of binary classifications that risk misdiagnosis, people around the border, for example, but we look at the trajectories, at these health trajectories, to see how individuals will progress fast, slow, or stable. So to show you an example, this is research cohort data before we get to real-world patient clinical data. This is data from different research cohorts, starting from ADNI, a large US cohort. And the classification that we ask the models to make is who is going to stay stable, who is going to develop, starting from MCI. And you can see that what the model gives us is a score that allows us to estimate how quickly people may move from that healthy, stable prototype And based on that score and based on normative data that we have, we can split people in broad categories, people that will remain stable, people that will slowly progress, and then people that will rapidly progress. This is done completely without kind of diagnostic labels, but we have the diagnostic labels because these are patients that have been diagnosed. And when we open the labels, you see that the people that the model says are moving quite fast towards Alzheimer's disease have already been diagnosed with Alzheimer's. Early Alzheimer's patients fall within the slowly progressive. No dementia patients, the model says they are stable. And the most interesting is this is data from research cohorts that they are pre-symptomatic. No symptoms, patients volunteered for research, run through the whole battery. And what you see is some of these patients remain stable, some of them, the model says, are progressing slowly. A very interesting space potentially for treatments. So the next step is what happens if we don't have labels? If we want to go 10 to 15 years before any symptoms occur, people don't have diagnosis. If people in their 40s, 50s, they would not want to have invasive testing done on them. We need to build what we call unsupervised models. These are models that can look at the data, learn patterns without having any labels. And we looked at these trajectory modeling approaches that then allow us to look at the path that people will take over time. Because we don't have clinical labels, we cannot do cross-sectional work. These are all healthy individuals. We work with ASNI, we work with UK Biobank in this case. And what these models do is they cluster individuals. They learn patterns across this multimodal space of different data types, and they cluster people. So here what you see is, the first graph didn't appear, but you would see nicely the clusters that appear. The model brings these trajectories, it learns the relationship between beta amyloid and gray matter, and you would see nice clusters here. I'm sorry they don't appear. And then, as I said, we don't have labels. Let me hide this for a minute. We look at our cognitive decline. We take a standard MMSE cognitive decline scale. And what you see is cluster D, which is one of these clusters, shows rapid cognitive decline. Cluster A shows no decline, and the other two clusters show a much slower decline. So these are the pathways, the projected pathways for these individuals. And then we look at this clustering, and of course we have labels because this is research data. We open the labels, and what you see is that this first cluster, which remains stable, has predominantly people that are healthy, while cluster D, which is the one that is declining the faster, has predominantly people that have already been characterized as having Alzheimer's disease. So these models give us the potential to do this without labels early on and allow us to build these modeling trajectories. So that's how we answer the question of how early can we predict. But that's not enough. We need to know what is that these models learn to be able to trust them. Otherwise, we cannot translate them into the clinic. And that's where we go into the core of the model and we interrogate the tensors. What is it in this space, the learning space of the model that the model learns? And what you see is, depending on what data we learn, we can now rank order the predictors that the model relies mostly in making the decisions. For example, if you put in biological data like gray matter, amyloid, APOE4, you see that a very strong contribution comes from amyloid. If you put in a little bit less invasive and cheaper data like what we would like to have in clinical practice, you see that, for example, MMSE, a basic scale, doesn't do very well for the model, but where the model gets more information is from more advanced, a little bit more elaborate tests like, for example, the ACE test and also gray matter. So by interrogating the models, we build them in a way that we can understand the mechanisms of the disease, we can understand interactions, and of course we can put in more interesting data here, for example, blood markers that are now coming up and inflammation markers, as we heard. How else can we interrogate the models? When we do look at these trajectories, we get scores. We do all sorts of mathematical statistical techniques like a scalar projection. We get scores that tell us whether a patient will remain stable, whether a patient will move away from healthy to disease, and how fast. These scores, then, we need to validate them with clinical variables, and that's what you see here. The score, this prognostic score we get from the algorithm, we show that it correlates strongly with cognitive decline, it correlates with increase in amyloid, it correlates with a decrease in gray matter, so with atrophy, and it also splits patients based on the APOE4 scales. So this is a validated marker against pathological markers, but it comes from the model, it carries the information, from all of these different modalities. So final challenge is, how can we make these models interoperable? How can we ensure that they generalize? They do not simply predict nicely in these research data, but actually can be applied in real-world clinical settings. And I'll show you example from working with team in CUMIN-MC, from working in Singapore with patient data, but also working on clinical trials for drug development. And so what you see here is data from CUMIN-MC. Team has the labels. We get the data, we get gray matter scores, and we get clinical scales. And we ask the algorithm, now the algorithm is trained, is a live engine, is completely trained. This is only test data, so we don't train the algorithm again, we just simply test it on this data, it's completely independent. And what you see is the model predicts that some of these individuals will progress rapidly, and these are the patients that have already been diagnosed with Alzheimer's. Patients already diagnosed with MCI, the model says are slowly progressing. Patients that have no AD but have other neurodegenerative, the model says they are rapidly progressive. Now, this is a model targeted to Alzheimer's, but we're now working at different dementia subtypes. Patients with simply functional, like attentional or memory symptoms that do not have disease pathology, the model says they are stable, they don't have Alzheimer's. Another, for example, stroke falls pretty much on the stable non-Alzheimer's, non-dementia space. Again, we don't have the labels, the model doesn't have the labels, the model just gets the test data and predicts. So for us, this was really exciting to see a true validation. As Tim explained, this data is not biased. The research data carries all sorts of biases, from demographics to genetics to everything that people that volunteer for research might be different from the population that, of patients that go to a memory clinic. Now, we moved on to another clinical patient population from Singapore because we wanted to see how the models behave in a completely different setting. And you see exactly the same process. Patients already diagnosed as AD, the model says they are rapidly progressive. Moderate MCI, closely to rapidly progressive, mild MCI, closer to slowly progressive, and healthy controls, the model says they are stable. Again, for us, this was a great, really, result to see in terms of validation in clinical practice and outside the UK. Now, this data offered us also the opportunity to ask the following question. That's a question I'm always asked. So if you were to put this today in a memory clinic, how long would you have to wait until the predictions of the models are validated? For patients, you will change over time, and will you have to wait for 3, 4, 5 years? This data set has longitudinal data. The patients have been followed for about 10 years. Here I show you in 6 years what happens. Again, what we do is we look at the different classes that the model gives us after re-stratifying the patients. And what you see is that the rapidly progressive is the red line. The model predicted that these individuals at baseline at time 1 will progress very fast, and that's what we see when, through a survival analysis, when we open the data over 6 years, they move very fast compared to the stable patients that they show the green flat line, no change here over 6 years as predicted by the model, and in between you see the slowly progressive patients, again as the model predicted from baseline, from first assessment, now being validated longitudinally. So for us, these methods really give us great hope that we can move forward to actually changing the clinical pathway. As patients come into the memory clinic at the first assessment, we can make a prediction of whether they are stable, they are slowly progressive, rapidly progressive, and really assign the patients to the right diagnostic pathway. Do they need a PET scan or not? Do they have comorbidities and they need to be looked into a different clinical pathway, diagnostic pathway? Do they need to be immediately hospitalized and potentially enrolled in interventions or treatments? I think I missed my clinical trial slide that I really wanted to show you, but these slides were made to the last minute, where we looked at clinical trial data and we show how the model can really stratify patients for clinical trials and show potential effects into drug treatments, but that's more to come. I just want to close by saying that we really hope that these tools will help clinicians to assign the right patient at the right pathway at the right time, will facilitate clinical trials in terms of the correct patient recruitment and stratification, and therefore facilitate their efficacy and pave the way to drug discovery. But most importantly, we hope that these models can really enhance patient well-being and reduce costs in healthcare systems. I'd like to close by thanking everybody who has contributed to this work. I'm really honored to be here on behalf of everybody, fantastic collaborators and early career researchers that dedicate lots of fun and effort to troubleshoot all of this data coming from maths, engineering, computer science, clinical neuroscience, memory clinics. I couldn't really list them all. Thank you.